Hello everyone. Let's compare a monohybrid cross to a dihybrid cross. In a monohybrid cross, we are concerned with one gene. And so let's say we're going to call this gene A to represent height. For gene A, you have two alleles because you have two parents. So you have one from your mom and one from your dad. You would then meet somebody who would also have two parents and so would also be diploid. And let's say that is their alleles. And so when we do this, we have to segregate alleles and put them in a Punnett square. Like this. And so when we segregate, we'll say that this is the mother and this is the father. We'll put the mom's alleles here. First, we'll put one of the segregated alleles here, and we'll put the other here. These represent her eggs. Notice that there's only one allele, which means instead of being diploid, they are haploid. Then we take the father's, and he makes these sperm that have these genotypes, and they are also haploid. Then egg and sperm meet together, and they create zygotes, all of which here would be heterozygous. And so that is a monohybrid cross. Now let's compare that with a dihybrid cross. We are still dealing with diploid organisms, and so we'll still have gene A represented like this. But now we are dealing with two sets of genes. That is what di means. And so instead of just A, we'll talk about gene B. And maybe in this instance, B would represent hair or baldness. B for baldness. If you have a capital B, you are bald. And so here we have a bald female that's really tall, and we have a short male who has hair. And so what we have to do here is we still segregate alleles. We still have to make haploid. We still have to make haploid sperm and egg. And so let's do that. And so, to be haploid means you only have one copy of every allele. So for example, I could get a capital A or a lowercase a from the mother. But then there's only one option from the father, and that is uppercase B. From the mother. That's all she has. Then if we go over here to the father, he only has allele A, little a. And he only has allele little b. And so this is the only possible sperm he can make, and these are the only possible eggs that she can make. And so then we do a Punnett square just like before, where we will put mom here, her eggs that are haploid, and the father's sperm here, which is also haploid. And then we will make the diploid zygotes in the middle. So let's start with the mother's eggs. And now we will put the father's sperm. We then combine those. And these are the possible genotypes that could result in their children. To check yourself, make sure that there is one copy of every allele in the parent's gametes, their sperm and egg. If there's more than one copy, you have made a diploid sperm, and that just does not exist. Also, make sure that every gene is present. You cannot create viable sperm or viable egg and have a child if your genes are missing. And so that's it. We have our results. That is how a dihybrid cross works.